Greetings. I am Captain Tsirk. In previous videos I addressed the modern scientific model of DNA, and its importance for biological life forms. In this video I will discuss the idea of genetic mutation, or changes in the genomic sequence. A genetic mut can be defined as sudden and spontaneous changes in the cell. Mutations are caused by radiation, viruses, transposons and mutagenic chemicals, as well as errors that occur during meiosis or DNA replication. They can also be induced by the organism itself, by cellular processes such as hypermutation. Mutation can result in several different types of change in DNA sequences. These changes can either have no effect, alter the product of a gene, or prevent the gene from functioning properly or completely. Due to the damaging effects that mutations can have on genes, organisms have mechanisms such as DNA repair to remove mutations. Mutations can involve large sections of DNA becoming duplicated, usually through genetic recombination. These duplications are a major source of raw material for evolving new genes, with tens to hundreds of genes duplicated in animal genomes every million years. Most genes belong to larger families of genes of shared ancestry. Novel genes are produced by several methods, commonly through the duplication and mutation of an ancestral gene, or by recombining parts of different genes to form new combinations with new functions. Here, domains act as modules, each with a particular and independent function, that can be mixed together to produce genes encoding new proteins with novel properties. For example, the human eye uses 4 genes to make structures that sense light, 3 for color vision and 1 for night vision. All 4 arose from a single ancestral gene. Another advantage of duplicating a gene is that this increases redundancy. This allows one gene in the pair to acquire a new function while the other copy performs the original function. Other types of mutation occasionally create new genes from previously non-coding DNA. Changes in chromosome number may involve even larger mutations, where segments of the DNA within chromosomes break and then rearrange. For example, 2 chromosomes in the Homo genus fuse to produce human chromosome 2. This fusion did not occur in the lineage of the other apes, and they retain these separate chromosomes. In evolution, the most important role of such chromosomal rearrangements may be to accelerate the divergence of a population into new species by making populations less likely to interbreed, and thereby preserving genetic differences between these populations. 2 classes of mutations are spontaneous mutations, molecular decay, and induced mutations caused by mutagens. Spontaneous mutations on the molecular level can be caused by different factors such as when base is changed by the repositioning of a hydrogen atom, altering the hydrogen bonding pattern of that base resulting in incorrect base pairing during replication. Another spontaneous mutation is called slip strand mispairing or denaturation of the new strand from the template during replication, followed by renaturation in a different spot, therefore slipping. This can lead to insertions or deletions. Induced mutations on the molecular level can be caused by chemicals such as hydroxylamine, base analogs, and alkylating agents. These agents can mutate both replicating and non-replicating DNA. Each of these classes of chemical mutagens has certain effects that then lead to transitions, transversions, or deletions. Nitrous acid converts amine groups on A and C to diazo groups, altering their hydrogen bonding patterns, which leads to incorrect base pairing during replication. Another kind of induced mutation may result from radiation. 2 nucleotide bases in DNA, cytosine and thymine, are most vulnerable to radiation that can change their properties. Mutation rates also vary across species. Evolutionary biologists have theorized that higher mutation rates are beneficial in some situations, because they allow organisms to evolve and therefore adapt more quickly to their environments. This has been an overview of genetic mutation. In future videos I will further address changes in DNA such as genetic engineering and species hybridization. Thank you for watching. Goodbye.